Alrighty, so in today's episode, we're going to continue talking about protein synthesis. Remember, in the last episode, we talked about the fact that protein synthesis occurs in two different stages. The first of which is transcription. So we took the code in DNA and translated it or transcribed it into RNA. Well, now the next piece, this is called translation, and we're taking that RNA code and translating it into proteins. So, translation, the process of making proteins from RNA. So we already made our RNA code in transcription, and now we're going to take this RNA code and we're going to actually put it into proteins. We're going to make proteins out of it. So, some important vocabulary when we're talking about translation. Our big player here is tRNA, and that's transfer RNA. So what the tRNA is doing is it's taking that code from the mRNA and actually pairing it up with an amino acid. So our transfer RNA here, it will match with an mRNA strand. And what the tRNA is they are short pieces that have 3 bases attached on one end, and then they have an amino acid on the other end. What happens is we also have what's called a codon. So the codon, these are on our mRNA. And a codon is simply 3 bases. So those are your letters, right? Your A, your T, your C, your G. So it's 3 of those in a row. So I might have A, T, T on my mRNA. That ATT is a codon. And this ATT is going to match up with a piece of tRNA that has an amino acid on it. Now, remember, amino acids are the monomers that make up proteins. Proteins are the polymer. And so I take lots of amino acids, I put them together, into a polypeptide chain. And this chain folds into a protein. There's only one D in folds. All right, so that chain will fold into a protein. So let's go step by step through this process. So I start out with a tRNA anticodon. So what the tRNA looks like, sort of this weird glob like this. And there are 3 bases here. Base, base, base. And on this end, I have an amino acid. So this is my tRNA. So my tRNA here, I have what's called an anticodon. It just means it matches up with a codon, which will match up with a codon on an mRNA strand. So here is my mRNA strand with lots of bases on it. 1, 2, 3. This is my anticodon here. Oops. this is my codon here. That's my codon. And depending on those letters will tell me what I actually have. What amino acid I have. So my tRNA up here binds with my mRNA strand, my codon, matches up with a codon. Then my amino acids up here will attach. So my amino acids are up here on one end of my tRNA, and then that anticodon is on the other end. So I get my binding first, then my amino acids that are on the other end will bond together. Then these amino acids up here will form a polypeptide chain, and it is this polypeptide chain that turns into a protein. So let's take a look, just so we can kind of see it. All right, notice here we have, these are our codons. So each one of these is a codon. Here is our tRNA molecule. Up here is an amino acid on the one end. I have a codon that says UUC. So if I use my matching rules, G bonds with that C, A bonds with that U, A bonds with that U, boom. So this matching anticodon will bond with that codon. Then this anticodon will bond with this codon. And then these two amino acids up here will form a polypeptide bond. And we now have multiple peptides together. Now, how do I figure out which amino acid goes? I need to use this chart. So what I'm going to do is read on the left-hand side first. See, it says first base. Let's go up here. A. My first base is an A. Then I read up top where it says second base code. My second base is a U. Here we go, U. My last base is my G. Those are the ones on the right-hand side here. Notice how I've got 4 lined up. So each one of these lines up with that third base pair. That's how I determine it. So G is last in my codon. So I have A, U, G. So I'm in box A, then U. So this box here is AU, AUG, right here, methionine. And I'm going to abbreviate it, MET. All right, let's do the next one. Over here, A, C, U, U, A, C, U, right here. 3 Let's try one more. A, G, C, right, A, G, C. Right here, serine. Let's do one last one just to practice again, because it doesn't have an A here. So U, G, G. U, we're in this box here because that's U, and then G, and then G here, tryptophan. Oh, there's a spelling error here. Tryptophan. All right, So that's how we build our amino acids to make our protein. One other thing I want to put out there. So this is one way you might see a codon chart. This is the other way. So let's just use GGG here. You might also see this. So say our code is GGG. What you do is you read from the inside out. So G is my first letter. G is my second letter. G is my third letter. And glycine would be the amino acid. All right, so thanks for listening to Lectures with Liddington, and let's all practice together.